Hi, good morning, dear students. Today we are discussing the 12th chapter, Biozoology, chapter number 9, Application of Biotechnology. Chapter number 9, Application of Biotechnology. The subtopic is recombinant vaccine and gene therapy. Recombinant vaccine and gene therapy. So, watch this video and take notes and refer your book. Okay, come to the topic, recombinant vaccine. What is vaccine? What is the definition of vaccine? Vaccine are the biological preparation. Biological preparation to produce active acquired immunity to particular infectious diseases. So once again, what is vaccine? These are biological preparation or microbial preparation, or the substances are extracted from the microbes like bacteria or viruses. Preparation to produce active acquired immunity. It is one kind of immunity can be developed inside the body to particular infectious disease. What is infectious disease? You know, the diseases are caused by microbes, are called infectious diseases. What is the recombinant vaccine? Recombinant vaccine are nothing but are produced by or developed by rDNA technology. rDNA technology. Recombinant vaccine are developed by or produced by rDNA technology. That means recombinant DNA technology, which inserting DNA encoding antigens into the body, which stimulate the immune response. What is the basic aim to use of the vaccine? Because which prevent the infectious diseases. But this is one of the new technique. This kind of vaccine is prepared by rDNA technology or recombinant DNA technology. So the recombinant DNA technology can be used to produce new generation of vaccine. New generation of vaccine. New generation of vaccine. Why it is a produce new generation of vaccine? Because the limitation of traditional vaccine production could be overcome by this approach. That is a new vaccine. That is to reduce the traditional vaccine. Traditional vaccine means the vaccine which are extracted in previous is old method. But this new vaccine are prepared by using what technique? rDNA technology. Recombinant vaccine are generally it is uniform quality. It is well quality because it protects the disease. It kills the pathogens in different ways and it produces less side effects than the other vaccine produced by some other methods. So what are the types of the vaccine? What are the three types of the vaccine? There are different types of the vaccine, but mainly there are three types. One is subunit recombinant vaccine. Second one is attenuated recombinant vaccine. Third one is DNA vaccine. I repeat, what are the three types of vaccine? Subunit recombinant vaccine, attenuated recombinant vaccine. The third one is DNA vaccine. Okay, what is subunit recombinant vaccine? The vaccine are used the components of pathogenic organism like bacteria or viruses. The components of the pathogenic organism rather than whole organism. Not we take the entire organism instead of a part or product of organism, pathogenic organism. What is a pathogen? We know that pathogens are infectious organism or disease producing germs or microbes is called pathogens. So this can be developed by, it is very suited for development of the rDNA technology that are very useful to new subunit vaccines. So what are the components which are produced by subunit? That means the product of the proteins. Another one is peptides. Next one is DNA of the pathogens. DNA of the pathogens. So subunit, listen again. Not the vaccine are prepared or extracted from not entire organism. The vaccine are extracted from their products. That products, it may be protein, is made up of proteins. It may be peptides. Peptide means what is it? These amino acids, one of the protein molecules. So the amino acids are linked by the peptides. So another one is directly DNA of the pathogen, not the whole organism. That is again, it is very important. The components of the, or it may be product of the pathogenic organism, not the entire organism. Suppose the bacteria which synthesizes protein. That protein alone we separate, or it may be peptides. So that is one kind of the proteins, the amino acid linked by the peptides are directly from the DNA of the pathogens. What are the advantages of the using of subunit recombinant vaccine? There are three advantages. One is purity in preparation. Purity in preparation. Another one is stability. The third one is safe use. Safe use. Purity in preparation. Because pure forms. Stability means it continues. It's stable form. Without changeable. So another one is safe use. So these are the three advantages. Next one is what? Attenuated recombinant vaccine. Attenuated means what? It is avirulent forms. It is not active form. Here the vaccines are prepared by genetically modified organism. That means GMO. Genetically modified organism. Either bacteria or it may be viruses. What is a genetically modified organism? Certain microbes are involved to changes its characters through the rDNA technology. They are called as genetically modified organisms. Certain bacteria are called genetically modified organisms. For example, Pseudomonas, some other bacteria which are used. But this kind of vaccine not used directly from the microbes. Because directly from microbes mean these are the virulent forms. These bacteria are harmful. These are harmful. These harmful bacteria or pathogenic bacteria become non-pathogen. The bacteria are pathogen. Naturally it is a pathogen because directly it causes disease. But these bacteria become inactivate or it become avirulent forms. Avirulent means what? Or non-pathogen means what? That microbes even enter inside the body, they do not cause disease. Any diseases. That form is called what is it? Avirulent. Many of the microbes which are living inside the body, but they do not cause any disease. Why? They are attenuated form. They are mild form. So they do not cause disease, but they can produce vaccine. They can produce vaccine. So they are called as attenuated form. Attenuated forms are avirulent vaccine. The pathogenic organism become non-pathogenic forms. The vaccine is extracted from that or separated from that is called attenuated. This attenuated vaccine is used as live vaccine. So these are called attenuated form. So it protects our body from the various infective organisms or pathogens which gives immunity. What is a DNA vaccine? The third type of vaccine, genetic immunization by using the DNA molecules. So directly DNA molecule. DNA means what is it? It is one of the genetic material of all organism, deoxyribonucleic acid. This is a DNA. Nucleic acid. It is a genetic materials. Directly the DNA molecule is inserted into the body to produce immunity. So this is one of the novel approach that came into 1990s. So that is a new method because the immune response of our body is stimulated by DNA molecule. I repeat, the immune response of the body is stimulated by DNA molecule. These are used in immunization process. What is immunization? Administration of the vaccine. Administration of the vaccine. So listen carefully, this is one of the novel method. Once the DNA is inserted into our body, immediately they synthesis antigen protein molecules. Antigen protein molecules. Once the gene are introducing, the genes are encoding. The genes are encoding antigenic protein. Antigenic protein. Encoding with the antigenic protein. This antigenic protein are inserted into the plasmids. Inserted into the plasmids. Into the plasmids. Again, for example, it is inserted into plasmid. Again, from the plasmid, what is called a plasmid? Plasmid is a circular DNA molecule of the bacterium. It is an additional circular DNA molecule in bacterium is called a plasmid. Plasmid here acts as a vector. Acts as a vector. Because the particular genes are extracted. These are the plasmids. I tell later that this is the plasmid technique. So listen here. But this is one example of the hepatitis B virus. Because the hepatitis B virus, the genetic material is DNA. They containing what is it? HB antigen protein. So this is HB antigen protein. HB antigen protein. What is HB? Hepatitis. Hepatitis, you know, it is one of the disorder which are causes. So hepatitis B antigen is present. Here it is one of the bacterium. Bacterium normally have two DNA molecule. One is the bacterial DNA, another one is circular DNA, is plasmid DNA. What is plasmid DNA? Additional circular DNA of some bacterium, so which are used as a vector. So immediately the plasmid DNA is removed from the bacterium, then cut out a small piece with the help of restriction endonuclease enzyme. Restriction enzyme. You know, the restriction enzyme are called as molecular scissor because it cut a specific site on the plasmid DNA, because this place is site for the attachment of the HB antigen protein. So these are recombined with this HB antigen from the viruses, recombined with the plasmid DNA to produce rDNA. rDNA are nothing but recombinant DNA. So this is the recombinant DNA. Here plasmid DNA along with another antigen producing genes are inserted. Here this rDNA inserted into the yeast cell. These are recombinant yeast cells. So recombinant yeast cells are, what yeast is normally used? Saccharomyces cerevisiae. This is one of the example of subunit. The first one is subunit recombinant vaccine. Hepatitis B vaccine. Hepatitis B vaccine is prepared by subunit recombinant vaccine. This is a general method, but it is one of the subunit recombinant vaccine. In the yeast cell, what yeast cell is used? Saccharomyces cerevisiae. Saccharomyces cerevisiae. Here, the yeast cells are used. This recombinant inserted, recombinant DNA is inserted. Again, it is inserted, multiply with the help of fermentation tank. These are multiplying enormous number. This is the multiplying recombinant yeast producing HB antigen in the fermentation tank. Fermentation means what? It is a tank which containing medium which multiply enormous number of the copies. Again, the recombinant DNA which produces HB antigens. This is the HB antigens. So this gene, particular genes immediately multiply to produces HB antigen. Again, that is extraction and purification of these antigens. This marketed in the HB antigens. These HB antigens are prepared by recombinant DNA technology. This can be used as vaccine against the hepatitis. So this is one of the example for that. That is what is the use of plasmids. So the plasmid acts as the vector. So immediately the gene into the target cells of the animal instruct to make the antigens. Suppose this plasmid DNA is inserted into the cell of the target animals. This gene make the cell to synthesize antigens. So antigens are the substances. So the gene which is synthesis to produce antigenic molecule, immediately our bodies produce what is it? Antibodies. So this is called as surface antigens. Displaced on the surface, our body synthesizes what is it? To secrete antibody molecules. Antibody responds to pre-floating antigens secreted by these cells. Listen carefully again. The gene, encoding gene which are inserted into the cells of target animal, that the DNA, that the inserted DNA instruct the cells to synthesize antigenic protein molecules displayed on the surface. So immediately our body to synthesize antibody. So the antibody react with or responds to the antigenic molecules secreted by these cells. So listen carefully. This is the gene which produces, to make the cell to synthesize antigenic protein. Antibodies react with antigens. So this is called as, this is a what type of vaccine? DNA vaccine. Why it is a DNA vaccine? Gene are inserted into the animals. So this kind of DNA vaccine do not cause any disease because it contains a few copies of the gene alone. What is the advantages of the DNA vaccine? Because it is inexpensive to produce and design the drugs. Okay, there are three kinds of the vaccine. So these three kinds of the vaccine have some advantages. What are the advantages? These three kinds of vaccine have some advantages. So one is it produces desired proteins, targeted proteins. Another one is it provides long-lasting immunity. And the third one is it a trigger. Trigger means what? Stimulate. Immune response to particular or a specific pathogens with less toxic effects. Listen, students. Commonly, the three type of the vaccine is produced by rDNA technology or recombinant technology. These are some common advantages. These are benefits which produces desired proteins to the animal. Suppose the DNA or gene are inserted into the body of animal or it is a cells, immediately they synthesize valuable proteins. Next one is it provides long-lasting immunity because long-term immunity for prolonged time. That kind of immunity which prevent the entry of the pathogens. And another one is trigger. Trigger means it stimulates immune response to specific pathogen. to particular pathogen with less toxic effect. This kind of vaccine is inserted, it do not cause the animal or it is the nearest cells or our own immune system. At the same time, it support our immunity power. This is one of the advantages. Next one is gene therapy. Another subtopic is gene therapy. What is the therapy means what? Treatment. You know that. What is the definition? It is one of the technique of rDNA technology, a technique of genetic engineering, rDNA technology or genetic engineering of faulty genes, replacement of faulty gene or defective gene, faulty gene by a corrective, healthy and functional gene. So it is a well-known definition part. Technique of genetic engineering of replacement of faulty gene. Faulty means defective gene. Defective gene by a corrective, healthy, functional genes. Or it is a simple definition: replacement of corrective gene in the place of defective genes. So this is one of the treatment, because most of the hereditary diseases that can be corrected by gene therapy method. Here the gene transfer into the person cells that carry one or more mutant allele. Mutant allele means mutation, mutated allele. So this gene therapy is a functional gene, is a healthy gene. That healthy gene or functional gene which produces normal phenotype. Disadvantages. For it is because this is the gene which are inserted into the cell. Inserted into the cell. The cell containing what is a defective gene. This part is a defective gene, defective part. This defective faulty part is replaced by normal gene. This kind of genes are called therapeutic gene. Therapeutic gene, or it is a healthy functional gene, because these cells are containing defective genes. This can be replaced. This can be corrected. So this gene therapy includes to correct some other hereditary diseases. Especially there are two kinds of hereditary disease. One is cystic fibrosis. Another one is hemophilia. What is cystic fibrosis? These are one of the hereditary diseases. It is a harmful effect which affect the lungs and the digestive system, because they can produce abnormal mucus, sweat. It becomes sticky and thick. So this is one of the defects of the lungs and the digestive system. What is a hemophilia? Hemophilia, otherwise called what is it? Lack of blood coagulation. This is otherwise called what? Bleeding disease. And so it is, the blood is normally clot after the injury, but in this case, the clot is not formed. It results in continuous bleeding is occur. So this is called as hemophilia. It is a well-known. These are otherwise called bleeder disease. It is a well-known hereditary diseases. So these can be corrected through the gene therapy. So the gene therapy method have two important strategies. Two important strategies. One is gene augmentation. Gene augmentation. Second one is gene inhibition therapy. Gene augmentation therapy. Another one is gene inhibition therapy. Okay, listen carefully. What is augmentation? Augmentation to produce, to make. This therapy involves insertion of the gene into the genome or insertion of the DNA or gene into the genome to replace missing gene products. It is a first method. Replacement of missing or defective gene products. That is the first method. It is the initial method. So suppose that person having defective gene which produces diseases, that person is identified and isolated the defective genes and inserted in to replace the defective genes. This is the first one, gene augmentation. Second one is called what? Gene inhibition therapy. Inhibit means what? To prevent, to stop. This therapy involves reduce or inhibit the expression of the dominant gene. I repeat, what is gene inhibition therapy? It involves insertion of antisense gene. Note it, antisense gene. What is the role of antisense gene? Antisense means what? Opposite. Suppose the second therapy involve to suppress or reduce insertion of the antisense gene. That genes prevent or reduce or suppresses the expression of the dominant gene. The nearest dominant gene is not expressed. So that antisense genes are used. The second method is gene inhibition therapy. The first method is replacement of missing gene product. The second method is insertion of antisense gene which suppress the expression of the or inhibit the expression of the dominant gene. Okay. Next, the types of the gene therapy. Here, types. There are two types of the gene therapy. One is somatic cell gene therapy. Second one is germline gene therapy. Same germline gene therapy. Okay, what are the two methods? Types: somatic cell gene therapy, germline gene therapy. What is somatic? Already we know. Somatic are the body cell. That is a non-reproductive cell. All body cells except the reproductive cells are called somatic cells. The somatic cell gene therapy involves introduction or insertion of the therapeutic genes into the body cells, into the somatic cells. That genes or therapeutic genes means what? It is healthy, corrective and expressive genes into the body. This kind of gene therapy involves to correct the single gene mutation. This corrects the single gene mutation. What cells are used in somatic cell gene therapy? Bone marrow, skin cells, etc. or bone cells. So the somatic cell gene therapy, when corrected, it do not change or transmit to the next generation because it is a non-reproductive cells. Any defect that occur, it is corrected only a person. It is not transmitted to another generation. It is not inheritable. The second one is called the germline gene therapy. What is germ? So this insertion of the genes into the sperm and the eggs. These are reproductive cells. This is the process or technique, insertion of the therapeutic genes or corrective gene, healthy gene into the sperm and egg. Sperm and egg what? It is the reproductive cells. Suppose this gene therapy when corrected in the sperm and the eggs, this defect do not change into the next generation. Suppose these are not corrected, this defect can be transmitted to another generation because the reproductive cells. Any defects that occur in the sperm and the egg, this can be inherited, but the somatic cell is not inherited. These are non-reproductive cells. So this, it is two advantages. One is the correction of the defective gene in the sperm and the egg or reproductive cell or germ cells. Another one is it prevents the disorder or defects that can be transmitted to next generation. Next generation. So this is germline gene therapy. Okay, the common method, the first clinical gene therapy. The first clinical gene therapy was in 1990 by French Anderson. So the first clinical gene therapy in the year developed in the year 1990 by the French Andersons in 4 years old girl. That girl is affected by adenosine deaminase deficiency. That is ADA. ADA means what? Adenosine deaminase deficiency. These are otherwise called SIDS. SCID, severe combined immunodeficiency disease. So adenosine deaminase, it is one of the enzyme which is needed from the bone marrow. Adenosine deaminase enzyme is essential because it stimulates the bone marrow stem cells to synthesize lymphocytes. What are the lymphocytes? There are B cell, another one is T cells. Listen here, adenosine deaminase, this enzyme is very important because it stimulates the stem cells of the bone marrow to synthesize lymphocyte like B cell, another one is called T cell. The B cell responsible for provide humoral immunity or antibody immunity. T cell responsible for the cell-mediated immunity. What in the case is occur? The adenosine deaminase synthesizing gene is defective. The adenosine deaminase synthesized are encoded by the gene because this enzyme is produced by gene. This information is carried by the gene. This gene is defective. What will be happen? The adenosine deaminase amount or secreting is very low. Or it is a deficiency. It is a nil product of the adenosine deaminase because of defective gene. Defective gene will not synthesize adenosine deaminase enzyme. It does not stimulate the stem cells of the bone marrow. So there is no production of the lymphocyte like B cell and T cells. These lymphocytes are very important to our body because which prevent the entry of the pathogen and kill the pathogens. So that girl or that person, it is a poor immunity power because both B cell and T cells are absent in this girl. It is a poor immune response or total death of the total decrease the amount of the lymphocyte. It affected by ADA deficiency. This is otherwise called SCAD. What is the treatment for this kind of disease? Bone marrow transplantation to the particular affected girl because the bone marrow to synthesis lymphocytes. Another one is directly the enzyme therapy. What is enzyme therapy? Adenosine deaminase enzyme therapy is given to the affected child. This is the first clinical child. It is corrected by the gene therapy. Okay. Thank you.